कोर्स कर दिए सर।
So we start the session. First of all, I welcome our today's speaker, Dr. Jyoti from IIT Delhi, and our participant, regular participants who have come on the time. So before handing over to the session, Jyoti, she is currently working as an assistant professor in the Department of Electronics and Communication at Nirma University, Ahmedabad. She has received her MTech degree in Electrical Engineering from IIT Gandhinagar in 2016 and PhD in Signal Processing from IIT Delhi in 2022. Following her PhD, she has spent 6 months as postdoctoral research associate at IIT Gandhinagar and next 6 months as assistant professor at National Forensic Sciences University, Gandhinagar. Apart from it, as I have interacted personally, Dr. Jyoti, she is very sincere in her work. While going to my supervisor, we both have had common supervisor, Professor S.D. Joshi. So she was very sincere and she secured first rank in her during her BTech time in the college. In during her MTech, she got 9.38 KGPA, which is very good. Again, during her coursework and the PhD, grade was 9.25. So overall, it's very showed the sincerity, dedication to the academics. And fortunately, she just 2 3 days before only I had conversation with her, and she readily accepted for the lecture session in our workshop. I'm really very grateful. So today she will discuss about the brain-computer interface and research, basically computational neuroinformatics and signal processing things. So handing over session to Jyoti.
Thank you so much. Thank you so much, sir, for the invitation. It is really a privilege for me to share my research work. So, sir, is my presentation visible?
Yes, sir. Perfectly visible.
Okay,
Graph
sir.
is and listen.
Yes, sir. Okay. So, my research area is mainly computational neuroscience. So, to understand the underlying dynamics of human brain. So, slowly we'll move to how we are doing this, but the major advantage of our work is we don't require any kind of training or testing. In real time we can analyze the brain signals using simple signal processing tools, techniques and mathematical concepts, and analyze those brain signals in order to find the underlying dynamics of human brain. So the content for my presentation will be, first of all, introduction, and then we'll see what is computational neuroscience. It is mainly having two parts. The first is brain modeling and the second is brain signal processing. So my research area is around brain signal processing, how we take brain waves and how do we process it. And then I will discuss 2 3 slides about preliminaries which is required to understand the fundamentals of our work, and then I'll be discussing my research work. So, if you have any doubt at any point of time or any suggestions if you want to give, I'm open to that. Anytime you can stop me and ask your doubts. So first of all, human brain is a complex network. So whatever we are doing, suppose now I am speaking to you, or we are gazing at stars, or we are eating, or whatever task we are doing. So different areas of our brain will be active, will be working together in order to carry out the desired action. So suppose we are writing. So now I am speaking, and suppose you are listening. So at that time, different areas of our brain will be working together to carry out the desired action. And by acquiring the brain waves, we try to understand the underlying dynamics, that how all these things are happening in our brain, what areas are getting activated, what signals we are getting, and how, even if we can diagnose about some neurological disorders, or if we can find out in resting states what is happening in our brain. So basically my work is around that understanding. So neuroscience nowadays, everyone, it neuroscience researchers are from different background. So electrical engineers, computer scientists, psychologists, doctors and neuroscientists, different areas, researchers from different background come together to understand the underlying dynamics of human brain. In particular, I am focused on computational neuroscience, in which we use mathematical models and computer simulations to understand the dynamics. So it is mainly having two parts. The first is mathematical modeling and the second is brain signal processing. What is exactly mathematical modeling? So now we are, everyone is talking about artificial intelligence. So obviously what a human is doing very naturally. Suppose if I ask anyone, if I say anyone, any student to go at the cafe and talk to his friend, so it will be very natural for him to go there and he will not think about anything. And suppose I want a robot to do the same task, so it will be very difficult for the robot to perform all this sequence. So he will have to leave from this room, and then he will have to go to stairs, and then search for his friend in the cafe, and all those things. So this sequence of events, if I want a robot to perform, it will be very difficult for a robot to follow all these tasks. So first of all, we will have to understand how a human brain is working, how we are performing a task. We have taken thousands of years to do whatever we are doing today. Suppose we take an example of cutting vegetables. So we took an years to understand how this thing is happening, and I want to do the same thing with a robot or artificially intelligent system. So first of all, we'll have to understand the underlying dynamics, how the brain is working. So mainly mathematical modeling consists of the theorist who tries to understand the underlying dynamics of brain, how the brain is working, and they come up with mathematical models of the same. And the second is brain signal processing. Brain signal processing is basically we acquire signals from the brain and we do analysis, we do signal processing approaches, we perform computer simulations and find the underlying dynamics, whether so if suppose it we are finding the neurological disorders in a particular situation. So, what is the need of modeling a brain? First of all, I'm talking about the first part, a brief introduction about the first part, and then we'll move to the second part, which is the research area of mine. So really, it sounds very interesting that if we can copy the human brain and if we can create an artificial intelligence system which is working very similar to the human brain, it sounds really interesting. But it requires too much understanding about our brain or neuroscientists. They are working on this area. How? So this picture is basically different neurons. So many neurons will be there. Billions of neurons will be there in our brain. Different neurons, they work together, they make connections within themselves and they make a larger network, and the whole network will work together in order to carry out any desired action. So for doing any task, the brain, different neurons, they will be firing together and they will be connected. So in the similar way, we can think of artificial neural network. So they have taken the idea from this fact that different neurons in our brain, they are working together to carry out the desired action. So in the similar way, in this slide we can say this is the biological neuron. So and the bottom one is the artificial neuron. So they are very similar. In biological neuron, if we'll see, the first of all dendrites will be there. So similar to that, we have inputs which will be summed here, and the same task is getting performed by biological neuron, which gets all the information from the previous neuron and then sum all the information, and firing will take place. So in the similar way, we have activation function here. So they are quite similar, inspired from the biological neuron. And similar to our complex brain network, artificial neural network is there, in which various neurons are there, artificial neurons are there, they are connected to each other and they have connections, and we carry out any action, suppose classification, regression, whatever we are doing or modeling, we are using the same thing. So basically, neuroscience and artificial intelligence, they are connected to each other. We make artificially intelligent models or different models inspired by human brain, and again we use ANN or artificially intelligent models or algorithms to understand the human brain. So this is just a two-way cycle. So so we can see an example. This is just an example of Tesla self-driving cars, in which they have designed a self-driving cars in which everything will be there. So this car will have so many sensors. So in which lane it will have the overall picture of the surrounding and how all these things are happening. So if a human would be sitting here and he will have to drive, so he will consider that what is there in the surrounding, in which lane I am driving, what are the nearby cars, how much distance should I be there. So all these consideration he'll take in account if a human driver will be driving that car. So considering all those facts, the Tesla self-driving car is a very good artificially intelligent system which is doing the same task, but it has certain limitations. Suppose a child is playing and his ball come in front of that car, so it will be very human can take the decision that what should I do, should I change the steering, or what should I do, should I directly stop. But it will be very difficult for a self-driving car to take the decision in that situation. So still AI is very far from reaching up to the human intelligence. The next is, nowadays a research article is there which claims that artificial intelligence systems are more effective than doctors at detecting breast cancer. So nowadays we are using artificial intelligence systems to do these things also. These are some of the examples how we are modeling the human brain and carry out the things which humans are doing. So this is just one another thing which is the inspiration of human brain. So suppose we are learning something, anything, if you are learning, so our brain will be rewiring. So if we are learning a thing over a period of time, so our brain, different neurons will rewire and make newer connections. So in the similar way, this is called neuroplasticity. Inspired by neuroplasticity, nowadays liquid neural networks are also there which consider this thing. So we are basically getting inspiration, we are understanding the human brain, we are getting inspiration from the human brain and applying in creating the mathematical model. So this is just one part which is theorist or mathematical modelers are doing. So now we come to brain signal processing. What is the role, what signal processing people are doing, or what computer scientists are doing with the brain signal in order to understand the human brain? So first of all, they acquire signals from the human brain. Human brain signals can be EEG, MEG, fMRI, different kind of imaging modalities are there. Through using that, we can acquire the brain signals. Then after signal acquisition is done, signal processing approaches or ML, different algorithms will be used in order to analyze that. So there are various situations. So there can be various application, such as in resting state. If someone wants to find out in resting state, when a person is not moving, not doing any task at that time, what is happening in our brain? So many thoughts. Even when we are sitting and we are not doing anything, in resting state also our brain will be active. Different thoughts will be there, and so many firing will be there in our human brain. So in resting state, if we want to find out, and you can use the resting state analysis in finding what is resting state when the person is not doing anything. So suppose a person is suffering from any psychological disorder and another person is normal. So in resting state, both of the person's brain state will be different. So we can use in order to diagnosis of any psychological disorder, in order to find how the in resting state they are working. So we can take an example of even seizure also in this case. And other than that, suppose depression, even it is claimed that if a person is in stress or depression, at that time the frontal lobe activation, which is this frontal area of our brain, that will be different in from a normal person, under the person who is under the stress. So we can do such kind of studies by acquiring brain signals. The second is while performing tasks. So this is having a critical applications in brain computer interface. So when we are performing any task, suppose I am doing some activity with my finger, motor tapping or something, or I am visually working on some of the activation. So how during different tasks my brain is responding, what are the brain signals? With brain signals can we identify what is happening and can we translate those signals into commands? So that comes under brain computer interface. The third is diagnosis, prediction, and analysis of neurological disorders. So these are various applications which researchers are doing with brain signals. So mainly EEG, MEG, fMRI, different imaging modalities are there which is used in order to understand the underlying dynamics of human brain. In particular, I'll be focusing on EEG because it is very economical also, and the temporal resolution of EEG is very good compared to other imaging techniques. And EEG, and in my work, as we'll go further, we'll see that I have worked on time varying brain signals. So this is the brief reprocessing pipeline. What do we do? So till now, anyone has any query? Should I stop, or how will you be asking questions, or if you have any query or anything? So anyone has worked in this area, EEG preprocessing?
So,
EEG?
so if anyone has a query, please type it in the chat box. Actually, here we have a provision of the chat box.
Okay, sir.
So
Okay.
I will then transfer to it.
Okay, sir. Okay.
Please feel free to ask the question. You can type in the chat box. The session should be lively. Never ever feel that we have to ask the question in the last or we have to wait.
Yes, you can ask anywhere, any doubt or any suggestions if you want to give. I am open to that. So, okay. So EEG is a non-stationary signal. As we know, it is not stationary. We cannot take the whole EEG data for 1 hour and we can work with that, because it is very highly non-stationary in nature. It is having too many artifacts. So suppose we are having a subject who is sitting and we have kept EEG cap on his head. So at that time, while acquiring the data, he may be having eye blinks artifacts will be there, heartbeat artifacts will be there, and muscle activity. Suppose he is moving his head or hand or whatever he is doing, power line artifacts will be there. So many artifacts will be there in our EEG data, but we just want to focus on what information lies in the EEG brain signals. And the brain signals will be very, the amplitude will be in microvolts, and in addition to that you will be having so many artifacts with your EEG data. So really it becomes very difficult to analyze that what is noise in that and what is EEG data. So first of all, if we have a little bit idea about what are these different artifacts, so we can just till some extent we can remove that. So suppose we take an example of eye blinks. So if eye blinks will be there, it is associated with lower frequencies, very low frequency, 0 to 0.5 hertz or so. So what we generally used to do, and the muscle artifacts, it is very high frequency, it will lie in the high frequency range, and the power line artifact will be around 50 or 60 depending on which where you have collected the data. So we generally perform filtering to remove these artifacts from our data. Once we have removed the artifacts from our data, it is not mandatory that every time we do all the pre-processing. These steps are optional. It depends on the situation where we want to use which artifact. So the next step is, generally some people used to do baseline correction. What is that? We remove DC offset. If any DC offset is there in your data, so we will take the DC, average, and we'll remove the DC offset from our data. And then re-referencing also. What re-referencing does, we take all the, so basically EEG, in EEG we have so many sensors which will be placed on our head. So suppose 64 sensors are placed on our head. So we take out of all the, we take average and we remove average from all the electrode signal. So what it does, if any noise, if outside any noise is there which is having similar impact on all the sensors, so we can remove those kind of noise using re-referencing. And some of the situation you will see that the your sampling rate, we should be also very careful that what is the sampling rate of our EEG acquisition system. Some of the EEG acquisition systems are working with 5000 hertz, that is the sampling rate of that. So those many samples, it depends on the situation what you want to analyze. So suppose I'm working, I'm only interested in alpha range, which is from 8 to 12 hertz. Generally EEG varies from 0 to, as we know, delta, theta, alpha, beta, different rhythms are there, and that is gamma also. So there different frequency bands are there for every delta, theta, alpha, beta, and gamma. So suppose we are focused on only alpha range, so it lies from 8 to 12 hertz, right? So my maximum frequency is 12 hertz. So if I want to do the analysis, so according to the Nyquist sampling rate, ideally FI should be greater than 2 FM. So mainly to 24 hertz, and in practical scenarios 24 hertz sampling rate is sufficient for my analysis. But that is the theoretical thing. If but in practice, if we will see at least 10 times more than that, we consider. So maybe 240 hertz. So 240 samples per second are sufficient for my analysis. But even if you take higher than that, so 5000 samples per second is too much for the sampling rate. So generally what we used to do, it will always, like, your processing will take too much time if you are working with too many samples. So in some scenario, depending on, if we want to work with gamma band, then we will have to say suppose 100 is the sample, 100 samples, and the band is 100 hertz. So according to sampling rate, or if we will say 10 times of the maximum frequency, if you will see, it is suppose 1000 samples per second is sufficient. So 5000 is still too much. So we can just perform down sampling so that the number of samples will be reduced per second, and our analysis will be comparatively faster and computationally efficient. You don't require too many resources when you are doing the calculation. So downsampling can be done. It depends on the situation. All the steps are optional. It is not mandatory that we do all the steps. The next is segmentation. As I have already mentioned that EEG data is highly non-stationary in nature. So what we used to do, generally, we divide the whole EEG data into small segments where each segment is considered to be stationary. We consider that the segment is small or sufficiently small enough that the signal, the system or signal properties do not change significantly. That's why we are working with small segments. The next step is principal component analysis or independent component analysis. So using as we all know, principal component analysis means we will have the dominant directions, dominant eigenvectors, and we will project our data on the dominant eigenvectors, and then we will, so basically reduction kind of thing will be doing with PCA component analysis, principal component analysis. And independent component analysis in EEG signal processing will be used in particular if you want to reject the artifact. Suppose eye related artifact, if I want to remove, so my EEG data will be divided into different components, and I can remove the artifacts which is heart artifact and different artifact. For artifact removal we use ICA for. So here one thing is there, in while applying principal component analysis we always consider dominant eigenvectors. Generally we consider the top 2 or 3 dominant directions, dominant eigenvectors, and we think that the maximum variations or variance of our data will be across those dimensions. But we thought in our work, you will see that in our work we have modified this thing. We consider that why only dominant eigenvector should be looked, what information lies in the eigenvectors corresponding to small eigenvalues. So why we are looking only at dominant eigen directions, we should also consider different eigenvectors are there. So what are those representing? So we have explored, other than the dominant eigenvectors, we have also explored the eigenvectors corresponding to smaller eigenvalues. It will be clear in our work that what we have done, but the motivation comes from here. Why we are looking, why we are considering that only dominant directions are more informative but not the other ones? So we'll see. And the next is, once you have done all the pre-processing, then some people work on functional connectivity analysis, where they have make functional connectivity networks, and then different correlation, coherence, different connectivity measures will be applied on the data, and then it will be fed to any classifier and so. The second is rhythm analysis, like I have already mentioned, delta, theta, alpha, beta. So some people used to divide your data, EEG data, into different bands, and then they used to process it. The third type is event related potential analysis, in which so maybe you have heard about P300. So different terms are there. So P300 and 170. So generally we used to analyze. So this particular stimuli is shown to the subject. So what is happening, what is P300? So truth light detection, people used to claim that P300 is a marker for truth light detection and so. And then some people are using and applying AI ML algorithm on this pre-processed data in order to diagnose neurological disorders, in order to do the classification and predictions also. Nowadays, newer terms are also there, graph signal processing. Some people used to do graph signal processing. They form graph signals or networks from the EEG data, then those network processing is getting done. So nowadays, the graph signal processing tries to extend all the traditional signal processing approaches in the graphical domain. So graphical domain is very irregular. So how we can extend all the things like Fourier transform, filtering, and sampling, all these concepts, how they are getting extended into graphical domain, that will be covered in graph signal processing. So many people are working in this area. Then brain computer interface, some works on, once we have used that EEG data, how we can convert this EEG data into commands which can be given to any machine. So suppose any machines, we can take brain signals, we can translate it into commands and can be given to machine to carry out the desired action. So this is roughly covers what EEG pre-processing pipeline. So in particular, what we are doing, our work focuses on graph theoretic approach to understand the human brain. So we conjecture that brain signals can be characterized as a sequence of different brain states. So suppose I am sitting here, I am speaking. So first of all, I am looking at the slide, then I am looking that what is the content, then this is my occipital lobe of my brain is getting activated, then I am thinking about it, that what should I convey, then I'm speaking, so my mouth is also involved, so motor area of my brain is also getting involved. So we think that when we are performing different tasks, suppose I am doing finger tapping, or I'm talking to you, or I am watching something, so when we are doing different tasks, different states or different brain states will be there corresponding to different tasks. And what do I mean by brain state? Brain state means that duration or in which interconnection between brain regions do not change. So when I'm doing any task, so my different area of my brain will be connected together. There will be some kind of connections between my brain area. When I'm doing finger tapping, when I'm speaking, different areas of my brain will be working together. When I'm doing any motor activity, different areas of my brain will be connected together. So we hypothesize that when we are doing a particular task, and so brain will be corresponding to a particular brain state, and by brain state we means that the interconnections between brain regions do not change when we are doing that particular task. So different brain states basically corresponding to different interconnections in the brain, and we consider that it will, even we are in the resting state, then also the brain state will be changing continuously, slowly but over a period of time. So in our work we will be looking at how, just by applying mathematical concepts and signal processing techniques, we'll be looking at how different brain areas are working together, and how the brain states are changing with when we are changing the task. So these are the four contributions or four work which I'll be discussing in today's presentation. The first is, we hypothesize that so as I have already mentioned that when we are doing different tasks, our brain will be in different brain states, and by brain state means different interconnections will be there. So in order to understand this thing, what we have done in the first thing, we have shown phase stimuli to the four different kinds of phase stimuli to the subjects, and we have just observed the functional interconnections between their brain states. So different stimuli were shown, and what is the difference in the underlying brain state corresponding to the different stimuli that was observed in the first. So it is very useful in face recognition. The second is, once in this was the static thing. Now when we consider, suppose 8 minutes data is there or 10 minutes EEG data is there, so continuously my brain will be changing its states. So in the second part we have seen how the brain is changing its dynamics, how the brain state is getting changed over a period of time. So that has been explored in the second thing, and we try to address a problem of BCI, that when the brain state is changing, are we able to identify what brain states are there and when the brain state is getting changed. So this was done. The third is, we used very naturally how we can make a network from the EEG data, and we in real time we try to detect the seizure onset with the simple approaches. And the fourth is, we propose a graph distance measure which generally finds what is the distance between any two networks. So the some of the preliminaries which is required to understand my work, the first is graph signal processing. Basically what we will do, we will take brain signals, EEG signals, so many sensors will be there. So we consider the sensors or the EEG electrode as nodes in my work. So those electrodes will be nodes, and the interconnections, the connections, or will be the edges, will be representing the edges. And we have used various functional connectivity measures in order to find the edge weights. So one of the most widely known or widely used functional connectivity measure is Pearson correlation coefficient matrix, which is just if suppose two sensors are there, F1 and F2, two sensors are there on your frontal lobe. So what is the correlation between these two signals? So that will represent the connecting edge. That will be the edge weight for that particular edge. So that will be given by Pearson correlation coefficient matrix. There are various functional connectivity measures such as coherence, phase synchronization, phase leg index, and etc. Granger, causality, and other measures are there which will give you directional connections also. So information is flowing from which node to which node. So there are different measures. What are the difference between them? The information which you are collecting or which they are finding between two signals are different. So Pearson correlation coefficient just give the linear relation between any two electrodes, but coherence is a frequency domain measure, and Pearson correlation coefficient is a time domain measure. Phase synchronization will give you about what is the phase relation between any two electrodes. So there are different kinds of functional connectivity measures which acquire different information. Phase leg index is also another measure which remove the volume conduction effect from the EEG data analysis. So there is one thing, if so if something is there in frontal area, so the signals will flow from frontal area to even back of my head. So inside my brain continuously signal will be traveling to all the electrodes. So that concept is called volume conduction effect. So phase leg index is called it removes till certain extent, it removes the volume conduction effect. So depending on the situation, people chooses different functional connectivity measures to from the brain network. So nodes are the EG electrodes, and edge weights between them are different functional connectivity measures. So these are some other functional connectivity measures. So diffusion is one of them. Diffusion is very common. So suppose if we say information is there at one node, and then it is traveling from that particular node to nearby nodes or nearby areas. So diffusion is happening on the network. So this is very natural phenomena. This is equivalent to net diffusion equation, which is there in the physics. So network diffusion is also very similar concept as a diffusion. We know that fundamental solution of the diffusion equation is heat equation. So similar to that, heat kernel is defined in the network domain, which heat kernel matrix, which is given by this formula, is just finding what is the, so basically it is a fundamental solution of the network diffusion equation, which tells which gets the complete dynamics of the underlying system. So this can also be, heat kernel can also be used as a functional connectivity measure if we consider the diffusion phenomena on the network which is there on your brain. So basically we have considered that brain can be also considered as a complex network in which different areas are there and they will be working together, and what will be the interconnections between different areas, that will be changing over a period of time. So everything revolves around graphs and networks, and how we have formed the networks, we have used functional connectivity measures. Now, when graphs are there, so graph distance, obviously we will be talking about how much is the similarity between two, if two events are there. One is speaking and one is suppose motor activity. So in during different activities, in suppose in speaking, different brain areas are getting activated together. So different network will form. This will be a completely different brain state, and another is motor activity. So in this case, different interconnections, different brain areas will be working together. Another brain network will be formed, so which will be very different from the previous activity. So how much is the similarity between these two graphs? So in order to find the similarity between any two brain networks, the phenomena is graph distances, which find the similarity between any two networks. So the first is, there are various kind of graph distances which is defined in the literature. The first is graph edit distance. Graph edit distance is generally just find what is the, if we want to change this particular, the network 1 to network 2, how many structural changes should I do in order to go from network 1 to network 2? So that is graph edit distance, which will just give which node is, which edge is present here and which edge is not present here, there is no connection. So how many number of structural changes are required to do in order to make network 1 into network 2? So that is given by graph edit distance. The graph matrix, the second is graph matrix distance. It talks about so network one, in this have, in this network one, basically network graph, they are similar thing. So different nodes are there, there will be some interconnections between them. So we, and interconnection is nothing but edge weight matrix, what is the relation between any two networks or how they are related. So I can represent that edge weights using a edge weight matrix, that matrix. And the second network will also have edge weight matrix. If we want, if we find the direct distance between edge weight matrices, that will give me matrix distance, what, how much is the different than edge weight matrices. The third is spectral distance. For one network and another network, we find the eigenvalues and eigenvectors, and based on eigenvalues and eigenvectors, we define a certain distances, that how much similar those two networks are, underlying networks. The fourth is feature based distance. In feature based distance, we take some features from network 1, we take some features from network 2, and then we define that how much dissimilar these two networks are. So these, there are several graph distances matrices in the literature which tries to find the similarity between any two networks. The third is, this is a very new phenomena which says it is say it is basically characteristic response vector. It is actually, so when any matrix is there, suppose we have edge weight matrix, so with that matrix we can obviously find any matrix, for any matrix we can find the eigenvalues and corresponding eigenvectors we can find out, right? So once we have the eigenvalues and eigenvectors are there, so CRV is basically a feature vector or feature vector which is combining the information of eigenvalues and eigenvectors and making a feature vector for that particular matrix. So how we are using it, how it is defined? First of all, how it is defined. So this is summation lambda I ui. So we take all the eigenvalues and eigenvectors, we take weightage sum of all the eigenvectors corresponding to their eigenvalues. So this is the characteristic response vector, which is defined in such a way that it is just a weighted sum of the eigenvectors. So basically for any matrix, so for one network we have one edge weight matrix. From that edge weight matrix we can find the eigenvalues and eigenvectors, and once we have eigenvalues and eigenvectors of that matrix, we can always form the characteristic response vector. So it was already defined that but the major limitation of this thing, as we have already discussed about something of PCA, suppose here correlation coefficient matrix of EEG data has been considered, and we have found the eigenvectors or and eigenvalues of the correlation coefficient matrix, and then we are finding this CRV, characteristic response vector, which is CRV. So yes sir.
Also, 1 minute. See, one question is there from
Yes, sir.
someone. See,
Yes,
how
sir.
to find distance between age-weighted matrix if we consider medical image? It
Age
even
weight.
yes, it is in
So
chat box also if you want.
okay, so directly matrix, the norm one, the norm two, the difference directly it is there. Norm two distance between any two matrices can be find. Norm any norm one, norm two, and several things are defined. So it is matrix distance basically between two different matrices. We find the difference and we find the norm of that. So that is Frobenius matrix distance. So many matrix distance measures are defined in the literature. Norm one, norm two, Frobenius, and so many matrix distances are there, which basically what it does, it takes matrix 1, it takes matrix 2 and find the difference and norm of that. Does it answer your question?
Someone, does it answer the question? Norm or norm two are the different ways to taking the distance.
Frobenius norm we have already studied, right? Matrix norms and
Yes, yes, actually it is there, but here actually in MNIT.
matrix norms we have studied, right? Frobenius norm, norm 1, norm 2, and so on.
Yes.
So we are just finding the difference and find the norm of that. So that's how matrix distance is defined in the literature.
Next question from someone is, can we use the CRE value for medical image?
Sure, sure. This is just a feature vector. We can use it anywhere. It was defined, it was actually proposed by one of my senior at IIT Delhi only. Siddharth sir
Yes,
has
yes,
defined
Siddharth
that.
Pamar.
Yes, sir. So he has defined this thing. This
Yes,
was proposed by him only in our one of our seniors in our lab.
yes, yes.
So should I proceed, sir, or any
Yes.
other questions? Okay. So basically the limitation of this CRV, as we have found out that it is giving lambda I, the max dominant eigenvector or dominant eigenvalue will have the maximum weightage in this summation. And if we will consider the less dominant eigenvalues or the eigenvalues corresponding to the eigenvectors corresponding to very small eigenvalues, that will be given very less weightage. So what is there in the less dominant eigenvectors? What information lies in that? Are they not meaningful? Are they not having any information, or just simply we should discard them, or is something like that like lies there in that less dominant eigenvectors? So from here our motivation starts. So why only we are looking at dominant eigenvectors? Even this CRA gives maximum weightage to dominant eigenvectors. So what lies in the lesser dominant eigenvectors? We wanted to explore that also. In our work we have seen, and even we have explored the all the eigenvectors corresponding to correlation coefficient matrices. And surprisingly, the result was completely different, which we will see. So, sir, if questions will be there, you will be like letting me know about that. So, otherwise, I should proceed.
Sure. Sure.
Okay, okay. So the limitation of this CRV is the maximum weightage is given to the dominant eigenvector. So in order to remove these limitations, we have done certain work. So now what contribution or what we have done further, we will see. So the first is importance of contrast feature. All the application, there are different applications which we will be, which I'll be briefing here. So different applications are there. So the first application, but the approach is same. Approach is graph theoretic approach. We are using brain signals, we are applying graph theoretic approaches, we are forming brain networks, and then we are using it in different applications. Though the first application is face recognition. So in this case, what we have done, actually I should start from here. So this is the experimental protocol. So it was collected the data by my guide Tapan Gandhi sir at MIT. So in this thing, this data, the 4 different type of stimuli for 30 different subjects have been shown to the subject. So this is a positive phase. This is the chimeric phase in which eye is there and the surrounding area of eye is preserved. This is the chimeric phase. The name was given as chimeric phase. The third is negative phase, and the fourth is only eyes. There is a difference between only eye and chimera phases. Only eye doesn't have the surrounding or it is not preserving the polarity around the eye area. But this chimeric phase is saving and having the contrast relationship around the eyes. So we just wanted to know during when different type of images for the same subject is shown to the subjects, whether different brain networks are formed or the networks which is getting formed in our brain are similar or are they different. So this was the experiment. It was done for 9 subject, total 128 electrodes were there, and sampling frequency was 500 hertz. For all the images, 30 images for each type of stimuli were presented in random order to the subjects. And what we have done, so this the question was asked, how do we find the matrix distance between any two networks? So this is what we have done in the first work. We have, so for this particular positive phase, we have acquired EEG data, and then we, for 30 times positive phases were shown, right? So for 30 times positive phases, we have accumulated the data, find the average data, and for that average data, we found, so that average data for that so I guess for that average data for positive phases, we have formed the functional connectivity matrix using Pearson correlation coefficient matrix. So in the 4 cases, case 1, case 2, case 3, case 4, in all the 4 the situation, we have formed functional connectivity matrices using correlation coefficient matrix. So now I have total 4 matrices for positive phase, chimeric phase, negative phase, and only eyes. Now, to find the distance among functional brain networks, we have used graph matrix distance, which is already existing distance. How do we find it? W corresponding to the network 1 and W dash corresponds to network 2. So this is how we find the matrix distance between any two networks. This is given by the distance. So we find out that there is a significant difference in the metrics in the networks which is getting formed in all the 4 scenarios. It was observed that positive phases and chimeric phases, the distance between them was very small compared to the distance between um, negative phases and only eyes. So we have also plotted the mean Pearson correlation coefficient matrix, which is plotted here. So the first one is for positive phase, this is for chimeric phase, this is for negative phase, and this is for only eye situation. So here we can see. So obviously, if we will look at lower left and upper right side of these 4 images, so they are mean Pearson correlation coefficient matrix for all the situations. We see in positive phases there is no. So here yellow shows the maximum correlation or 1 between 2 different electrodes. So here on x-axis we have different electrodes. So from 0 to 128, on y-axis also we have different electrodes, and this is just plotting the correlation coefficient matrices. And the color shows 0. So we have taken the absolute value of the correlation coefficient. So that's why the range is from 0 to 1. 1 shows the maximum correlation, 0 shows no correlation. So here we can see in positive phases there is no correlation or no activation in this area. In chimeric phases, a little bit correlation we can see in the diagonal off diagonal elements. In negative phases and only eyes, we can see very high correlation in the lower left and upper right side. So we have found out that the electrodes corresponding to this, the 100 to 128 and this is 1 to around 40 electrodes were corresponding to frontal lobe of the brain. So they were placed on the frontal area. So when person were looking at positive phases in frontal area, there was the electrodes were not too much correlated with each other. Even in chimeric phases also very small correlation was there, but in negative phases and only eyes there were strong correlation. So we have also plotted corresponding brain networks in that situation. So here we can see this is the considering all the edge weights, and this the second figure considers only off diagonal elements, only if we plot the off diagonal elements of the human brain. Even in this case, we can see that in frontal lobe area, this upper part shows the frontal lobe. So in frontal lobe, there is not too much connections between different electrodes. But here, if you will see, there are too much connections in the frontal lobe area for negative phases in only eyes. And even just we have plotted the off diagonal elements, we can see that positive phases have very small correlation between frontal electrodes, and chimeric phases a little bit more, and negative and only eyes, there is too much correlation between different area. So we have found out that when a person or a subject is looking at the positive phases, so the this is very, so it is very easier to recognize, right? We can recognize positive faces very easily. So the cognitive demand or how much processing I am doing, that will be lesser because I am easily recognizing that particular face. But if only negative face is presented or only eyes are presented, it will be very difficult to recognize, and difficult recognition, so cognitive load will be more. If cognitive load is more, so the connection between different frontal regions will be too high. That's why in negative phases and only eyes, there is too much correlation between different areas, because they are working and trying to find out what is this phase, they are trying to recognize. And chimeric, if at the end, if you will see chimeric phases, so even it is not that much like only eyes and negative phases. So a little bit cognitive task is happening, a little bit of thinking is there in order to recognize chimeric faces, but that is not too much. So from this study, we can conclude that yes, contrast relationship around the eyes also plays a crucial role in identification or recognizing any face. So even in corona times, or sometimes we used to put mask on our face, but other person can easily recognize us because the eye area and the uh, surrounding area is there. So with that also we can find. So this activity or this work just generally captures why different brain network areas are there, what is their correlation, and how do we interpret it. So this, but the limitation of this work, or what we have done in this work, is we have just observed static brain networks. So we have not talked anything about how with time the networks are changing, how whether they are changing. In this whole work, we have considered that brain networks are static, but this is not the situation. Ideally, my brain networks will be changing over the period of time, they are not static. So I will have to consider the non-stationarity of EEG signals also in while doing the analysis. So by inspired from this thought, we have proposed time varying brain networks. With by what do we I mean by time varying brain networks? Over a period of time, the brain networks will be continuously changing, and we just want to see from one state to next state how my brain is getting transition, whether it is moving slowly or how it is doing the transition between different brain state corresponding to different tasks. So in order to do this also, we have taken several EEG signals real time. All this data is real time EEG data, which has been taken from some of the data was there with us in our lab, and some of the data is publicly available. So we can also you can also explore the data set. So basically, in this second task, person or subject will be doing different activities, and how in a particular activity how the brain state is there, and when the activity is getting changed, whether we are able to track that or not, how we are able to recognize all these things, brain state transition. One more thing, we have not done any kind of training on any data in my work. We have used the data in real time. Only the present sample will be present seconds data will be compared with the previous second data. And based on this continuous analysis of the EEG signal, we come up with taking the transition of brain state. So it is having very, see, computation, the no training testing time consuming, it will not be there, it works in real time. So we have worked for different applications in real time, and it was working. And hardware also will be required very less, because we don't need any memory at all, because current second data will be compared to the previous second, and then based on that we are taking the decisions in our thing. So this is the just a screenshot of our EEG data. This is a continuous EEG data. It is non-stationary in nature, so it will be divided into small segments. And we consider that this segment is, and this frame is sufficiently small enough so that functional interconnections or the connections between brain region do not change significantly in this particular frame. And then next frame is there, so they are sufficiently small enough so that we can also see how slowly the brain states are changing over a period of time. So in this first thing, we have divided it into small segments. For each segment of data, we have formed functional connectivity matrices using correlation coefficient matrix. So for frame 1, we have correlation coefficient matrix, from frame 2, from frame 3, and for all the frames, we have functional connectivity matrices, not at the first end. So as suppose this much data is there with me, I'll make it, I will work with it, and I will find the correlation coefficient matrix. Then next second, I'll get the next data and I'll make the functional correlation coefficient matrix. So like that, we are working on a small segments in real time. So, acha, one more thing here, we have used overlapping segments, not discrete. Overlapping segments are there. Why overlapping segments? So that we smoothly captures the transition. It should not be direct, like in frame 1 state 1 was there, in frame 2 state 2 was there. So we didn't want that type of transition. We wanted to see how slowly my brain is changing its state. So we have taken overlapping EEG data segments for the whole analysis. Now, so I have already mentioned a little bit about that. Principal component analysis says that dominant eigenvectors represent the direction of maximum variations. So generally we project in the direction, on the directions of maximum variance, and we generally discard and think that smaller eigenvalues and eigenvectors will be just noise, will be there. So we consider that as dominant eigenvectors represent the direction of maximum variance, similar to that less dominant or the least dominant the eigenvectors corresponding to smallest eigenvalues will be representing invariance eigenvectors, or and minimum variance eigenvectors, or the direction which is having minimum variance. So minimum variance means it may also represent the invariant information of the system. So we have observed that dominant eigenvectors represent maximum variance directions, and least eigenvectors are, we have termed them as the invariant eigenvectors, which have the directions which represent the directions of minimum variance. So if we want to capture something which is static in nature in our brain, or some static information is there, or some invariant information of the brain, if we want to capture, ideally we should look at the eigenvectors corresponding to smaller eigenvalues. So we have seen that. So and then CRV was there. CRV was giving a maximum weightage to the dominant ones, and there was very less weightage given to the invariant eigenvectors. So now onwards, I will be using these two terms. Dominant means the eigenvectors corresponding to larger eigenvalues, and invariant eigenvectors means the eigenvectors corresponding to smallest eigenvalues will be them. So we have formed subband CRV. Subband CRV is what, instead of summing all the eigenvectors all together weighted with their eigenvalues, you choose a continuous block of eigenvectors which will be relevant in that particular situation. So in my situation, suppose I see that I'm just thinking about, I'm not considering about how much eye blinks are there in a situation. Suppose I'm doing a motor activity which is continuous for 1 minute. So I just wanted to know, for this 1 minute some motor activity is happening. So this is the static information which is not very significantly changing. So we should focus on, for this static information or invariant information of my brain, we should look at the invariant eigenvectors of the brain. So we have devised subband CRV, which is basically taking only a block of eigenvectors weighted with the eigenvalues. So instead of summing all the eigenvectors, we can just choose the eigenvectors which will be having the relevant information. I'm not saying that always we should consider the dominant ones or the least dominant ones. It depends on the situation which is having the information. They all are having some information, but in which information we are interested in that particular application. So in this situation, we wanted to capture the invariant information or static information of the brain state. So we have focused on invariant eigenvectors. Then we have formed the subband CRV vectors, which is obviously lambda I ui summation lambda I ui. But in this case, instead of summing all the eigenvectors, we are just summing the eigenvectors corresponding to smaller eigenvalues. We have taken 4 or 8 some smaller eigenvalues and those corresponding eigenvectors found a weighted sum. So that was the subband CRV in this situation. We'll see there are different applications which I have seen, and somewhere I have used the dominant ones, somewhere I have used the least dominant ones. So it depends on the situation. If we are just trying to find out what is the suppose we want to plot our data where maximum variance is there, we should focus on the dominant ones. In some scenario we are just finding what is the invariant thing which is happening, what is the static thing. If we want to capture the static information, we should focus on the eigenvectors corresponding to smaller eigenvalues. So what we have done, this is the first data set in which for 1 minute, uh, so this is the 8 minutes data, 8 minute easy recording for 20 participants, total 72 electrodes were there, and 1 minute eyes open, 1 minute eyes closed, like that the experiment was designed, eyes open, eyes closed for 1 minute. So continuously the brain state was changing. After 1 minute, for 1 minute the eye state was eye open. So while eye open, I just wanted to capture what is there. So this is a simpler experiment in which no typical tasks are there, but the second is multitask experiment. So there also we have applied the same thing. So what we have done, we have EEG recording for this whole 8 minutes. Then the whole EEG data is divided into small overlapping segments. For each segment, we have functional Pearson correlation coefficient matrix. From correlation coefficient matrix, we found all the eigenvectors and eigenvalues. Then we have for all the matrices, and then for every functional connectivity matrices, or for every network, if I say, so we have eigenvalues and eigenvector, and for each frame we have formed one subband CRV, which is just summation lambda IUI with invariant eigenvectors. So like that, for all the frames we have one subband CRV, which is subband characteristic response vector. Now we have just found the Euclidean distance between consecutive subband CRVs. So this is no training, no test, no nothing, no learning. You take first minutes data, first seconds data. Suppose I think 1 or 2 second is the frame size in this situation. For 1 second we have the data, we found subband CRV for this 1 second frame. The next data is coming, for that we found the subband CRV for the next frame, and we are just finding the Euclidean distance between consecutive subband CRVs. And just by finding the subband CRVs between consecutive frames, we have come up with these results. What these results are showing, so the first is characteristic response vector. If we take all the eigenvectors, all 64 eigenvector, where the maximum weightage is given to the dominant ones and the least is given to the less dominant ones. So if you take that, and if you find the distance between consecutive CRV and subband CRV, there is a difference between those. So CRV consider all the eigenvectors, subband CRV considers only a block of eigenvectors in which we are interested in. So we'll have to see the physical interpretation of what is the physical meaning or what they are representing. What is the information? Yes, sir.
So see, you have already a 1 hour session, so we can have a break for 10 minutes.
Okay, sir. Okay.
At 4:15 we will resume.
Okay, sir. Okay.
Okay.
Okay. Thank you, sir.